Welcome to Sleeping Researcher, your gentle guide to the wonders of science, narrated softly to help you drift into peaceful rest. Here, we explore fascinating scientific ideas and discoveries told in a calm and soothing way to ease your mind and carry you gently towards sleep. It's been 4 weeks since I started on this journey. There's been 14 subscribers as of this video. The channel have also been raking in hundreds of views. It's not a lot, but I am grateful for every single one of you. Tonight, we embark on a quiet journey into the world of gene editing, an extraordinary field where science is reshaping the very fabric of life. Imagine being able to rewrite the instructions that make up living organisms, to correct genetic errors, or to enhance traits, all with precision and care. This is the promise of technologies like CRISPR, along with other remarkable tools that are transforming medicine, agriculture, and our understanding of biology. Before we begin, if you enjoy this gentle exploration of science, please consider liking the video and subscribing to Sleeping Researcher. Your support helps us continue sharing these calming stories from science, designed to soothe your mind and enrich your dreams. As we settle into this story, allow yourself to relax and imagine the possibilities unfolding quietly in laboratories and research centers around the world. Together, we will discover how gene editing is shaping the future of life, one delicate change at a time. Let us begin. Imagine a world where the very blueprint of life, the DNA, can be gently rewritten, corrected, or enhanced with remarkable precision. This is no longer a distant dream, but an emerging reality, made possible through the advent of gene editing technologies. Among these, CRISPR stands as a beacon of innovation, offering scientists the tools to target specific sequences within the genome and make deliberate changes. Unlike traditional genetic modification methods, which often involve trial and error and unpredictable results, CRISPR and similar techniques provide a level of control that was once unimaginable. By harnessing naturally occurring mechanisms found in bacteria, these technologies have opened a new chapter in biology and medicine, where the code of life can be edited with unprecedented accuracy and efficiency. The implications extend far beyond the laboratory, promising applications that could alleviate genetic diseases, improve crop resilience, and deepen our understanding of life itself. Gene editing is not a single technology, but a collection of innovative methods that have evolved rapidly over recent years. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is arguably the most well-known tool due to its simplicity and effectiveness. It works by guiding molecular scissors, known as Cas proteins, to precise locations within the DNA strand where cuts can be made to remove or replace genetic material. Other technologies, such as TALENs and zinc finger nucleases, also enable targeted DNA modifications, but often require more complex design and engineering. The beauty of these systems lies in their ability to precisely edit genes without affecting unrelated areas of the genome, thereby minimizing unintended consequences. This precision is crucial as it allows researchers to explore genetic functions with greater clarity and develop therapies that directly address the underlying causes of diseases. As gene editing techniques continue to mature, their potential applications grow ever more diverse and impactful. In medicine, these technologies hold the promise of correcting mutations responsible for inherited disorders such as sickle cell anemia or cystic fibrosis, potentially offering cures where none existed before. Beyond human health, gene editing is also transforming agriculture by enabling the development of crops that can withstand environmental stresses, resist pests, or provide enhanced nutritional value. These advancements could play a vital role in addressing global challenges, including food security and environmental sustainability. Moreover, gene editing fosters new avenues for scientific research, allowing us to better understand the complex interactions within genomes and the molecular basis of life. The journey from fundamental discovery to practical application continues to unfold, illustrating the profound ways gene editing is shaping the future. The ethical and societal dimensions of gene editing are equally important as the technological breakthroughs themselves. With the power to alter life's fundamental code comes a responsibility to consider the implications of such changes. Discussions surrounding the use of gene editing in humans, especially germline modifications that could be inherited by future generations, emphasize the need for careful regulation and thoughtful deliberation. Scientists, ethicists, policymakers, and communities worldwide are engaged in conversations aimed at balancing innovation with safety, equity, and respect for natural diversity. This ongoing dialogue ensures that gene editing advances not only with technical excellence, but also with an awareness of its broader impact on humanity and the environment. As we continue to explore these frontiers, gene editing stands as a testament to our ability to understand and influence the intricate mechanisms of life itself. At the heart of modern gene editing lies a remarkable discovery known as CRISPR, an acronym for clustered, regularly interspaced, short palindromic repeats. Originally found as a natural defense mechanism in bacteria, CRISPR allows these microorganisms to recognize and cut the DNA of invading viruses with remarkable precision. Scientists have ingeniously adapted this bacterial system to become a powerful tool for editing genes across a wide range of organisms. By guiding a special enzyme called Cas9 to specific sequences of DNA, CRISPR enables targeted cuts and subsequent modifications to the genetic code. This method is not only more accurate than previous gene editing technologies, but also simpler and more cost-effective, opening a new era in genetic research and biotechnology. As we delve deeper into how CRISPR functions, it becomes clear that this technology is transformative because it harnesses a natural process, repurposing it to rewrite the very instructions that govern life itself. The elegance of CRISPR lies in its specificity and adaptability. Researchers program a small piece of RNA, known as the guide RNA, to match a precise DNA sequence they wish to edit. When introduced alongside the Cas9 enzyme, this guide directs Cas9 to the exact location on the genome, where it creates a double-strand break. the cell's own repair mechanisms then take over, either disabling a gene or allowing the insertion of new genetic material. This level of control is unprecedented, enabling scientists to explore gene functions, correct mutations, and develop potential therapies for genetic disorders. Beyond human medicine, CRISPR holds promise in agriculture, where it may help create crops that are more resilient to climate change, pests, and diseases. In environmental science, gene editing could assist in controlling invasive species or preserving endangered ones. The versatility of CRISPR continues to inspire innovations that extend far beyond its original discovery. CRISPR is part of a broader family of gene editing technologies that are continually evolving. Before CRISPR, tools like zinc finger nucleases and TALENs allowed targeted DNA modifications, but were more complex and costly to design. CRISPR's relative simplicity has democratized gene editing, making it accessible to more laboratories and accelerating scientific progress. However, this rapid advancement also brings challenges, such as off-target effects, where unintended parts of the genome might be altered. Researchers are actively developing new variants of CRISPR and complementary techniques to enhance precision and reduce risks. These refinements aim to make gene editing safer and more reliable for future clinical and ecological applications. As the technology matures, it invites a careful balance between innovation and responsibility, ensuring that its power is wielded with the utmost care. The profound capability of CRISPR to edit genes with unprecedented accuracy reshapes our relationship with biology. It offers a glimpse into a future where diseases caused by genetic mutations could be corrected before they manifest, where crop yields might increase sustainably, and where biodiversity preservation could include genetic rescue strategies. Yet, the journey from laboratory discovery to real-world application requires rigorous testing, ethical oversight, and public engagement. Each step forward is measured not only by what can be achieved, but also by what should be pursued. The dialogue surrounding CRISPR and gene editing is as much about hope and possibility as it is about caution and respect for the complexity of life. In contemplating this, we recognize that gene editing technologies like CRISPR are not just scientific tools, but profound catalysts for reflection on the future of life itself. The story of CRISPR-Cas9 begins within the microscopic world of bacteria, where it serves as a natural defense mechanism against invading viruses. Scientists observed that certain sequences of DNA, known as clustered regularly interspaced short palindromic repeats, or CRISPR, were interspersed with snippets of viral DNA. This pattern suggested a form of genetic memory, allowing bacteria to recognize and disable viral invaders more efficiently in future encounters. Paired with Cas proteins, particularly Cas9, this system could precisely locate and cut viral DNA, a discovery that revealed an elegant molecular tool for genetic targeting. This foundational understanding laid the groundwork for reimagining CRISPR-Cas9 beyond its natural role, inspiring researchers to harness its precision for editing genes in other organisms. The realization that a bacterial immune system could be repurposed as a customizable gene editor was both unexpected and revolutionary, opening new doors for molecular biology and biotechnology. The breakthrough moment arrived in 2012 when Jennifer Doudna and Emmanuelle Charpentier published their seminal paper demonstrating that the CRISPR-Cas9 system could be programmed with a simple RNA guide to target virtually any DNA sequence. This innovation transformed CRISPR from a natural curiosity into a versatile and accessible genetic engineering tool. Unlike previous gene editing methods, which were often complex, inefficient, or limited in scope, CRISPR-Cas9 introduced a simplicity and flexibility that democratized gene editing research. It empowered laboratories around the world to edit genes with unprecedented ease, speed, and precision. This discovery was more than a technical achievement. It represented a paradigm shift in how scientists could manipulate the very code of life. The elegance of this system lies in its adaptability and its potential to rewrite the genetic instructions that underlie health, disease, and biological function. Following the 2012 breakthrough, the landscape of genetic engineering evolved rapidly. Researchers began applying CRISPR-Cas9 across diverse fields. From correcting mutations responsible for inherited diseases to developing crops with enhanced nutritional value and resilience. The technology's ability to target specific genetic sequences enabled more refined approaches to gene therapy, minimizing unintended effects and improving safety. Concurrently, other gene editing technologies such as TALENs and zinc finger nucleases continued to advance, each contributing unique strengths and considerations. Yet, CRISPR's ease of use and efficiency rapidly established it as the preferred tool, catalyzing a wave of innovation and discovery. The pace of progress underscored both the promise and the responsibility inherent in wielding such powerful molecular tools. Beyond its scientific and technical implications, the discovery of CRISPR-Cas9 sparked a profound ethical and philosophical dialogue. Questions emerged about the boundaries of human intervention in natural processes, the potential consequences of editing germline cells, and the equitable access to these transformative technologies. The conversation extended beyond laboratories and regulatory bodies, engaging the broader public in reflections on our shared future with biotechnology. This interplay between innovation and introspection highlights the evolving relationship between humanity and the living world, reminding us that each leap forward invites careful consideration of its broader impact. In this light, the discovery of CRISPR-Cas9 stands not only as a milestone in science, but as a catalyst for ongoing exploration of our values, responsibilities, and hopes for life's future.
To appreciate the profound impact of CRISPR-Cas9, it helps to understand the elegant simplicity of its mechanism. At its core, CRISPR functions much like a pair of molecular scissors, guided by an exquisitely precise GPS system. This system allows it to seek out and target specific sequences within the vast expanse of genetic material that makes up an organism's DNA. The process begins when a specialized molecule, known as the Cas9 enzyme, pairs with a carefully designed RNA guide. This RNA sequence is complementary to the DNA segment researchers want to edit, enabling Cas9 to navigate the genome with remarkable accuracy. Once the RNA guide locates its matching sequence, Cas9 binds tightly to the DNA and creates a precise cut at that location. This targeted break in the DNA strand is what sets the stage for subsequent genetic alterations. The simplicity and precision of this mechanism have revolutionized how scientists approach gene editing, transforming a once cumbersome and imprecise endeavor into a highly accessible technique. After Cas9 makes its incision, the cell's natural repair machinery takes over. This biological response to DNA damage is central to the versatility of CRISPR-based technologies. Cells typically repair double-strand breaks through pathways such as non-homologous end joining or homology-directed repair. In the first, the cell rejoins the DNA ends, but often introduces small insertions or deletions which can disrupt the function of a targeted gene. Alternatively, if a repair template is provided, homology-directed repair allows for precise replacement or insertion of genetic material. This feature enables researchers not only to disable faulty genes, but also to correct mutations or insert new sequences, expanding the possibilities for therapeutic intervention. The interplay between CRISPR-induced breaks and cellular repair pathways creates a powerful toolkit for rewriting the genetic code, allowing for modifications that were previously unimaginable. This elegant harnessing of natural processes exemplifies the harmony between engineered tools and biological systems. Beyond Cas9, the CRISPR toolbox continues to grow, incorporating variations of the enzyme with distinct properties and functions. Scientists have discovered other proteins within the CRISPR family, such as Cas12 and Cas13, which expand the range of possible gene editing applications. Cas12, for example, targets single-stranded DNA and has been adapted for highly sensitive diagnostic tools, while Cas13 specializes in RNA editing, enabling temporary modifications without altering the underlying genome. These advances illustrate the adaptability of CRISPR systems and their potential to fine-tune genetic information at multiple levels. The ongoing development of these molecular instruments promises to expand the boundaries of what is achievable, ranging from correcting genetic diseases to enhancing agricultural resilience and beyond. Each new iteration invites deeper understanding and careful stewardship of this powerful technology. As we explore the mechanics of CRISPR and its expanding repertoire, it is important to remember that this technology is but one branch of a broader family of gene editing tools. Earlier techniques, such as zinc finger nucleases and talons, laid the groundwork by enabling targeted DNA modifications, albeit with less ease and flexibility. The emergence of CRISPR-Cas9 marked a paradigm shift due to its simplicity and efficiency, dramatically lowering barriers to entry and accelerating research worldwide. Yet the principles underlying these methods share a common thread: the precise and intentional alteration of genetic sequences to better understand and ultimately improve living organisms. This lineage of innovation reflects the cumulative efforts of countless researchers seeking to unlock life's code with ever greater finesse. As we continue to delve into the mechanics and possibilities of gene editing, it becomes clear that CRISPR is both a product of and a catalyst for this ongoing journey. Though CRISPR-Cas9 has garnered much attention for its groundbreaking capabilities, it is important to appreciate the broader landscape of gene editing technologies that have contributed to our understanding and manipulation of the genome. Before CRISPR's rise, tools such as zinc finger nucleases, or ZFNs, provided a means to target specific DNA sequences with remarkable precision. These molecular scissors are composed of engineered protein domains that recognize particular DNA triplets fused to a nuclease that cleaves the DNA. Designing these proteins required intricate knowledge and laborious customization, making the process time-consuming and technically demanding. Nevertheless, ZFNs demonstrated the feasibility of targeted genome editing and set the stage for subsequent innovations. Their ability to induce double-stranded breaks at chosen locations allowed cells to repair DNA in ways that could correct mutations or introduce new genetic information, opening doors to potential therapies and agricultural improvements alike. This early chapter in gene editing history reminds us that progress often builds upon the foundations laid by persistent efforts to solve complex biological puzzles. Alongside zinc finger nucleases, another notable technology called transcription activator-like effector nucleases, or TALENs, emerged as a versatile alternative for genome editing. TALENs harness proteins derived from plant pathogens, which naturally bind DNA in a modular fashion, recognizing one base pair at a time. By assembling these modules in specific sequences, researchers could tailor TALENs to target virtually any DNA region with high specificity. This modularity simplified design compared to ZFNs, although TALEN construction still required considerable effort and expertise. Like their predecessors, TALENs induce targeted double-strand breaks that stimulate cellular repair pathways, allowing precise genetic alterations. Their development highlighted the creativity and resourcefulness of scientists in adapting natural biological systems for human benefit. While TALENs may have been somewhat overshadowed by CRISPR's rapid adoption, they continue to offer valuable advantages in contexts where CRISPR may face limitations, such as targeting certain genomic regions or minimizing off-target effects. This diversity of tools enriches the gene editing toolkit, enabling tailored approaches to varied scientific questions and therapeutic challenges. Beyond these well-established methods, the field of gene editing is continuously evolving,
with novel techniques that expand our capacity to rewrite genetic information. Emerging technologies explore new mechanisms such as base editing and prime editing, which aim to make precise single base changes without inducing double-stranded breaks. These refined approaches promise to reduce unintended consequences and improve safety profiles for potential clinical applications. Meanwhile, advances in delivery systems, from viral vectors to nanoparticles, seek to bring these molecular tools into cells and tissues more effectively and gently. Each innovation contributes to a rich tapestry of strategies designed to overcome the complexities of living systems. The accumulation of these diverse methodologies reflects a collective aspiration to understand life at its most fundamental level, and to harness that knowledge for the betterment of health, agriculture, and the environment. As we consider the spectrum of gene editing technologies beyond CRISPR, we glimpse a future where precision and adaptability will guide our interaction with the genome in increasingly sophisticated ways. In recent years, the quest for enhanced precision in gene editing has led to remarkable advances that refine the ability to target specific sequences within the vast expanse of the genome. Early gene editing techniques, while groundbreaking, often carried the risk of off-target effects, unintended modifications that could disrupt vital genes or regulatory elements. Today, scientists employ sophisticated guide RNA design algorithms and engineered nucleases with improved specificity to minimize these risks. For instance, variations of the CRISPR-Cas system, such as high-fidelity Cas9 enzymes, have been developed to recognize target DNA with greater discrimination, thereby reducing the likelihood of collateral damage. This heightened accuracy is not merely a technical achievement, but a critical step toward translating gene editing from laboratory experiments into safe, effective therapies for human diseases. As the molecular tools become more precise, the potential to correct genetic mutations that cause inherited disorders grows ever more tangible, offering hope to millions worldwide. Alongside improvements in enzyme specificity, the advent of base editing and prime editing technologies has revolutionized the scope of possible genome modifications. Unlike traditional CRISPR systems that rely on creating double-stranded breaks, these newer methods enable single nucleotide changes with minimal disruption to the DNA backbone. Base editors chemically convert one DNA base to another, while prime editors employ a reverse transcriptase coupled to a modified Cas protein to write new genetic information directly into the genome. These innovations reduce the risk of unintended insertions or deletions and significantly lower the chance of triggering cellular damage responses. The elegance of such precise editing mechanisms lies in their ability to mimic natural DNA repair processes, guiding cellular machinery to accept corrected sequences without activating alarms. This refined control opens pathways for treating complex diseases, where subtle genetic changes can restore normal function without compromising genome integrity. Equally important to the success of gene editing
is the development of efficient delivery systems capable of transporting molecular tools safely into target cells. Viral vectors, with their natural ability to infect cells, have been engineered to carry gene editors while minimizing immune reactions and toxicity. Meanwhile, non-viral approaches such as lipid nanoparticles and engineered extracellular vesicles offer versatile platforms for transient, controlled delivery. These carriers can be tailored to reach specific tissues, cross biological barriers, and release cargo in response to environmental cues. As delivery technologies improve, they not only enhance the precision of genome editing by limiting exposure to non-target cells, but also reduce the potential for off-target effects linked to prolonged editor activity. The synergy between precise editing enzymes and refined delivery vehicles is crucial for achieving therapeutic outcomes that are both effective and safe. The continuous refinement of gene editing tools reflects a deepening understanding of the intricate relationship between molecular machinery and cellular environments. Researchers are now integrating computational modeling, high-throughput screening, and deep sequencing technologies to predict and monitor editing outcomes with unprecedented resolution. Such comprehensive analyses allow the identification of even rare off-target events and inform iterative improvements in both enzyme design and delivery strategies. This meticulous approach underscores the commitment to ensuring that gene editing interventions do not compromise genomic stability or cellular function over time. By embracing this thoroughness, the scientific community moves closer to realizing gene editing therapies that balance innovation with the highest standards of safety. The promise of gene editing technologies, such as CRISPR-Cas9, extends deeply into the realm of medicine, where the potential to correct genetic mutations at their source offers profound hope for patients with inherited disorders. Conditions like sickle cell anemia, which arise from a single nucleotide mutation affecting hemoglobin structure, have long presented challenges due to the complexity of managing symptoms and preventing complications. Recent advances, however, demonstrate that by precisely targeting and repairing the faulty gene in hematopoietic stem cells, it is possible to restore normal function to red blood cells. This approach not only addresses the root cause, but also circumvents the need for lifelong treatments or repeated transfusions. Clinical trials have begun to show encouraging results, with patients experiencing reduced symptoms and improved quality of life following ex vivo gene editing and autologous transplantation. The ability to intervene at such an early molecular stage transforms the treatment paradigm, shifting from symptom management to curative intent. Cystic fibrosis, another devastating genetic disorder characterized by mutations in the CFTR gene, exemplifies the breadth of gene editing applications in medicine. Unlike sickle cell anemia, which affects blood cells, cystic fibrosis primarily impacts epithelial cells in the lungs and digestive system, leading to thick mucus buildup and chronic infections. Targeting this disease through gene editing requires innovative delivery methods capable of reaching affected tissues efficiently and safely. Researchers are exploring viral and non-viral vectors that can transport gene editing components directly to lung tissues,
enabling correction of the mutation responsible for impaired chloride transport. Early preclinical studies reveal that restoring proper CFTR function can alleviate the defects underlying the disease, paving the way for treatments that could dramatically improve pulmonary health and reduce the burden of infections. This work highlights how gene editing is not confined to blood disorders, but extends to complex organ systems where precise genetic repair can restore physiological balance.
Beyond specific diseases, the refinement of gene editing technologies continues to open new therapeutic avenues for a wide array of genetic conditions. The use of base editors and prime editors, which allow for single-letter changes in the DNA sequence without creating double-strand breaks, further enhances the precision and safety of these interventions. Such advancements are especially important in treating diseases caused by point mutations, where even subtle modifications can restore protein function. By reducing the risk of unintended genomic alterations, these tools offer a path towards safer, more predictable gene therapies. Moreover, the integration of patient-derived induced pluripotent stem cells with gene editing provides personalized models for testing and refining treatments before clinical application. This synergy between technological innovation and individualized medicine exemplifies the future direction of therapeutic development. While the clinical benefits of gene editing in fighting genetic diseases are becoming increasingly apparent, the challenges of delivery, immune response, and long-term efficacy remain areas of active investigation. Researchers are optimizing delivery vehicles, such as lipid nanoparticles and engineered viral vectors, to enhance tissue specificity and minimize immunogenicity. Simultaneously, long-term follow-up studies are critical to monitor the stability of gene correction and ensure that edited cells function properly within the complex environment of the human body. Regulatory frameworks and ethical considerations guide the responsible translation of these powerful technologies from bench to bedside, emphasizing patient safety and informed consent. This cautious yet optimistic approach fosters a landscape where innovation can thrive alongside rigorous oversight, ensuring that gene editing fulfills its therapeutic potential sustainably. As gene editing therapies advance toward broader clinical use, the horizon expands beyond monogenic diseases to encompass complex conditions where multiple genetic factors contribute to pathology. The ability to modulate gene expression, either by activating protective genes or silencing harmful ones, broadens the scope of treatable diseases, including certain cancers and neurodegenerative disorders. In this evolving landscape, the integration of gene editing with other modalities, such as cell therapy and immunotherapy, creates multifaceted strategies to combat disease. The ongoing collaboration between molecular biologists, clinicians, and bioengineers continues to refine these approaches, ensuring that they are both effective and accessible. This dynamic interplay heralds a new era in medicine, where the precise rewriting of our genetic code becomes a cornerstone of healing and restoration. In recent years, CRISPR technology has emerged as a transformative tool in cancer research, offering unprecedented precision in editing the genetic material
of living cells. Unlike traditional therapies that often affect both healthy and malignant cells, CRISPR enables scientists to target specific genetic sequences that contribute to cancer's growth and survival. One particularly promising application is the engineering of immune cells, such as T cells, to enhance their ability to recognize and eliminate cancer cells. By modifying genes that regulate immune cell function, researchers can effectively reprogram these cells to seek out tumor markers with greater accuracy and persistence. This approach not only improves the immune response against cancer, but also reduces the risk of unintended side effects that can arise from less selective treatments. The potential to harness our own immune system, empowered by gene editing, marks a significant step forward in personalized medicine and immunotherapy. Central to this innovative strategy is the concept of chimeric antigen receptor T cells, or CAR T cells, which have been genetically altered to express receptors that specifically bind to antigens found on cancerous cells. CRISPR plays a vital role in streamlining and enhancing the creation of these CAR T cells, allowing for more efficient and precise modifications than previous gene editing methods. By knocking out genes that inhibit immune activity or inserting genes that improve tumor targeting, CRISPR-engineered CAR T cells exhibit increased potency and durability when deployed in the body. Moreover, this precise editing reduces the likelihood of immune rejection and off-target effects, which has historically limited the broader application of cell-based therapies. The synergy between CRISPR and cell therapy is opening new avenues for treating cancers that were once considered difficult or impossible to manage effectively. This evolving landscape demonstrates how gene editing can be seamlessly integrated with immunological insights to create smarter, more adaptable treatments. Beyond CAR T-cell therapies, CRISPR is also being employed to investigate the complex genetic underpinnings of cancer itself. By editing genes within tumor cells in laboratory settings, scientists can better understand the roles specific mutations play in cancer progression and resistance to therapy. This knowledge fuels the development of targeted interventions that disrupt these pathways, potentially halting the disease in its tracks. Furthermore, CRISPR allows for the creation of more accurate cancer models, mimicking the diverse genetic profiles found in patients. These models serve as crucial testing grounds for new drugs and combined treatment regimens, accelerating the path from laboratory experiments to clinical trials. The versatility of CRISPR thus extends beyond therapeutic applications, encompassing fundamental research that deepens our grasp of cancer biology and informs future innovations. As this research unfolds, it continuously informs the design of next-generation gene editing tools tailored to the unique challenges posed by cancer. The integration of CRISPR with other emerging technologies, such as synthetic biology and advanced delivery systems, further broadens its potential in cancer treatment. For example, researchers are exploring ways to engineer immune cells that not only identify and attack tumors, but also adapt to changes in the tumor microenvironment, overcoming mechanisms that cancers use to evade destruction. Nanoparticles and viral vectors are being refined to deliver CRISPR components, directly to target cells in the body, minimizing systemic exposure and enhancing safety profiles. These multidimensional approaches reflect a deeper understanding of cancer as a dynamic and evolving adversary, one that requires equally sophisticated and adaptable therapies. By combining gene editing with real-time monitoring and feedback systems, future treatments may be able to adjust their activity in response to the tumor's behavior, ensuring sustained effectiveness over time. This convergence of technology and biology exemplifies the forward momentum in designing smarter, more responsive cancer therapies.
As CRISPR continues to mature as a technology within cancer research, ethical considerations and rigorous safety evaluations remain at the forefront of its clinical development. Ensuring that engineered immune cells function as intended without unintended consequences is paramount, necessitating comprehensive preclinical studies and carefully designed clinical trials. Collaborations across disciplines and institutions help establish robust guidelines and monitoring protocols that protect patient well-being while enabling innovation. This careful stewardship fosters public trust and accelerates the responsible translation of gene editing breakthroughs from bench to bedside. With these foundations in place, the potential for CRISPR to reshape the future of oncology grows ever more tangible, setting the stage for the next wave of medical advances that will continue to refine our approach to combating cancer and other complex diseases. Beyond the realm of medicine, gene editing technologies like CRISPR are beginning to cultivate profound changes in agriculture, offering new avenues to enhance the resilience and productivity of crops. Traditional breeding methods, while invaluable, often require many generations to achieve desired traits. In contrast, precise gene editing allows scientists to target specific regions of plant genomes, accelerating the development of varieties that can better withstand environmental stresses. For example, by altering genes related to drought tolerance, researchers have been able to create crops that maintain growth even under limited water availability. This is particularly significant as climate change intensifies, making water scarcity a pressing issue for farmers worldwide. The ability to engineer traits such as pest resistance also reduces reliance on chemical pesticides, promoting more sustainable farming practices that protect both the environment and human health. These advancements are not merely theoretical. Field trials are already demonstrating how gene-edited plants can thrive in challenging conditions, hinting at a future where food security is bolstered by innovations rooted deeply in molecular biology. In addition to enhancing resilience, gene editing opens doors to improving the nutritional content of staple crops, addressing global concerns related to malnutrition and dietary deficiencies. By precisely modifying genes involved in the synthesis of essential vitamins and minerals, scientists can increase the nutritional value of crops such as rice, wheat, and maize. For instance, biofortified rice varieties with elevated levels of vitamin A precursors aim to combat vitamin A deficiency, which affects millions, particularly in developing regions.
This targeted approach to nutrition goes beyond traditional fortification methods by embedding beneficial traits directly into the plant's genetic code, potentially providing a sustainable and cost-effective solution for vulnerable populations. Moreover, these modifications can be accomplished without introducing foreign DNA, differentiating gene-edited crops from older, genetically modified organisms, and potentially easing regulatory and public acceptance challenges. As these innovations progress, they hold the promise of not only feeding more people, but nourishing them better, contributing to the overall health and well-being of communities worldwide. The pace of discovery in gene editing also supports the development of crops with improved yield, a vital factor in meeting the demands of a growing global population. By fine-tuning genes that regulate plant growth, flowering time, and resource allocation, researchers aim to optimize the balance between productivity and environmental sustainability. For example, modifying genes responsible for root architecture can enhance nutrient uptake efficiency, allowing plants to thrive in less fertile soils, while reducing dependence on synthetic fertilizers. This has broad implications for reducing the environmental footprint of agriculture and promoting more resilient food systems. Importantly, these technologies are being employed with a mindful approach to biodiversity and ecosystem health, ensuring that innovations harmonize with natural processes rather than disrupt them. Collaborative efforts between molecular biologists, agronomists, and farmers help ensure that gene-edited crops are developed responsibly and tailored to the specific needs of diverse agricultural landscapes. As gene editing tools continue to evolve, so too does the potential to integrate multiple beneficial traits into single crop varieties. This multiplexing capability allows for the simultaneous improvement of yield, stress tolerance, and nutritional quality, supporting more holistic agricultural solutions. The precision of CRISPR and related technologies reduces unintended genetic changes, increasing confidence in the safety and efficacy of edited crops. Regulatory frameworks around the world are gradually adapting to accommodate these advances, balancing innovation with careful oversight to ensure that gene-edited plants meet rigorous safety and environmental standards. These developments are fostering a new era in agriculture, one where science and stewardship come together to meet the challenges of a changing climate, evolving ecosystems, and the nutritional needs of humanity. As these promising strides continue, the dialogue between technology and tradition remains essential, guiding the responsible integration of gene editing into the fabric of global food production. Among the most intriguing advances in gene editing is the development of gene drives, a technology designed to bias the inheritance of specific genetic traits within wild populations. By ensuring that a particular gene is passed on to nearly all offspring, gene drives have the potential to rapidly spread desired modifications through entire species. This capability opens new avenues for controlling invasive species that threaten native biodiversity, as well as disease vectors such as mosquitoes that transmit malaria, dengue, and other illnesses. Imagine a future where populations of harmful insects could be gently nudged toward extinction or rendered incapable of carrying pathogens, reducing the burden of disease on human health without relying on chemical pesticides. However, the power of gene drives to alter ecosystems on a large scale also demands careful consideration. The irreversible nature of some gene drive applications means that unintended consequences could ripple through food webs and habitats, affecting species beyond the initial target. As a result, scientists and ethicists emphasize the importance of robust ecological assessments and containment strategies before any release into the wild. The ecological impact of gene drives extends beyond the immediate goal of species control, touching upon the delicate balance of ecosystems that have evolved over millennia. Invasive species often disrupt local environments, but their removal or alteration may create new niches or allow other organisms to flourish in unexpected ways. For example, reducing a mosquito population might benefit human health, yet it could also affect predators or competitors within the ecosystem. To navigate these complexities, interdisciplinary research combines genetics, ecology, and environmental science to model potential outcomes and identify safeguards. This collaborative approach helps ensure that gene drive interventions are designed with a deep understanding of natural interdependencies, minimizing risks while maximizing benefits. Moreover, the reversible or self-limiting designs of gene drives are being explored as a means to provide greater control and flexibility, offering a pathway to correct or halt modifications should unforeseen effects arise. Ethical considerations surrounding gene drives are as significant as the scientific challenges. The prospect of altering wild populations raises questions about human responsibility, consent, and the rights of ecosystems and future generations. Decisions about deploying gene drives require transparent engagement with diverse communities, including indigenous peoples, local stakeholders, and global policymakers. This inclusive dialogue helps to weigh the potential health and environmental benefits against cultural values and ecological preservation. It is a reminder that science does not operate in isolation, but rather within a complex social fabric where trust, respect, and shared goals are paramount. Regulatory frameworks and international agreements are evolving to address these concerns, promoting responsible research and stewardship of gene editing technologies that may cross national borders and affect shared environments. As gene drives continue to progress, from the laboratory to possible field applications, ongoing monitoring and adaptive management will be critical components of their responsible use. Scientists are developing sensitive detection methods and early warning systems to track gene drive spread and assess ecological responses in real time. This vigilance aims to provide opportunities for intervention if unexpected patterns emerge, ensuring that gene editing remains a tool of precision and care. In parallel, public education and transparent communication foster understanding and informed decision-making, helping to build societal confidence in these powerful technologies. Together, these efforts contribute to a future where gene drives may offer novel solutions to some of the most persistent challenges in conservation and public health. As the remarkable capabilities of CRISPR and other gene editing technologies continue to expand, they bring with them profound ethical questions that invite careful reflection. One of the most prominent concerns involves the concept of designer babies, where gene editing could be used not only to prevent hereditary diseases, but also to select for preferred traits such as physical appearance, intelligence, or athletic ability. This prospect raises important debates about fairness, equity, and the potential for exacerbating social inequalities. The idea of customizing human life challenges deeply held beliefs about natural diversity and the acceptance of human differences. It also provokes questions about consent, as future generations would inherit changes made without their agreement. These considerations urge society to carefully weigh the benefits of preventing suffering against the risks of creating new forms of discrimination or loss of individuality. As these discussions unfold, they underscore the need for robust ethical frameworks and inclusive dialogue that respect diverse perspectives and values. Beyond human applications, gene editing technologies also pose significant ethical dilemmas when it comes to biodiversity and ecological balance. The release of genetically modified organisms into the wild, whether for disease control or conservation, carries the risk of unintended consequences that may disrupt existing ecosystems. Altering genetic material in one species could ripple through food webs, affecting other organisms in unpredictable ways. This possibility prompts careful consideration of the precautionary principle, advocating for thorough risk assessment and cautious implementation. Moreover, the potential for gene drives to spread edited genes across populations raises questions about control and reversibility. How can we ensure that these powerful tools do not inadvertently threaten the richness of life on Earth? Addressing these concerns requires collaboration between scientists, ethicists, policymakers, and the communities most affected, fostering stewardship that honors both human progress and environmental integrity. The governance of gene editing technologies also presents a complex landscape in which ethical, legal, and social dimensions intersect. Different countries and cultures approach these issues with varying values, regulations, and levels of public engagement. This diversity highlights the challenge of establishing international standards that balance innovation with responsibility. Transparency, accountability, and public participation emerge as key principles in shaping policies that guide the development and application of gene editing. Engaging with indigenous knowledge systems and respecting cultural differences further enrich these conversations, ensuring that technological advancement does not override human dignity or community rights. In this intricate tapestry of considerations, ethical vigilance becomes as essential as scientific rigor in navigating the possibilities and perils of gene editing. As these ethical challenges continue to unfold, they invite us to reflect on the kind of future we wish to create with gene editing technologies. The balance between hope and caution, innovation and reflection will shape not only scientific progress, but also the fabric of society itself. It is a journey that calls for humility, empathy, and a commitment to shared values that transcend individual ambition. With thoughtful stewardship, gene editing may become a force for healing and harmony, rather than division or harm. This ongoing dialogue opens pathways toward a future where technology and ethics walk hand in hand, guiding us gently into new realms of possibility. Around the world, nations are carefully navigating the complex regulatory landscape that surrounds gene editing technologies, such as CRISPR. Different countries are adopting varied approaches to balance the exciting potential of these tools with the need to ensure safety, ethical oversight, and social responsibility. In many parts of Europe, for example, strict regulations emphasize precaution, often imposing rigorous review processes before any gene editing research or applications, especially those involving human embryos or germline modifications, can proceed. This cautious stance reflects deep-rooted cultural values and a commitment to protecting human dignity and environmental integrity. At the same time, it encourages transparent public dialogue and multidisciplinary collaboration, fostering a collective understanding of the implications of gene editing. Such regulatory frameworks, while sometimes perceived as limiting innovation, serve as guardrails to prevent unintended consequences and to build public trust, which is essential for the responsible advancement of biotechnology. In contrast, countries like the United States and China are exploring a more flexible regulatory environment that aims to stimulate scientific innovation while maintaining oversight. In the US, agencies such as the Food and Drug Administration and the National Institutes of Health have established guidelines that govern research and clinical applications, often encouraging innovation within defined safety parameters. This approach allows for rapid progress in fields such as gene therapy and agriculture, where gene editing has demonstrated promise in treating genetic disorders and improving crop resilience. China, meanwhile, has invested heavily in gene editing research and infrastructure, signaling a strong commitment to leadership in biotechnology. However, recent regulatory adjustments have underscored the importance of ethical considerations, especially following high-profile controversies involving human embryo editing. These developments illustrate a dynamic interplay between ambition and caution, where regulatory bodies strive to foster scientific breakthroughs without compromising ethical standards or public confidence. In other regions, including parts of Africa, Latin America, and Southeast Asia, regulatory frameworks are evolving in response to both global scientific trends and local needs. Many countries are establishing policies that emphasize capacity building, education, and regional cooperation to harness gene editing for public health, agriculture, and environmental sustainability. For instance, initiatives aimed at controlling vector-borne diseases through gene drives are being carefully evaluated to ensure ecological safety and community consent. These efforts highlight the importance of context-sensitive regulation that respects cultural diversity and prioritizes equitable access to emerging technologies. By fostering inclusive governance and encouraging international dialogue, these regions contribute valuable perspectives to the global conversation on how gene editing can be deployed responsibly and justly. International organizations and scientific bodies also play a crucial role in shaping the regulatory landscape by developing harmonized guidelines and ethical frameworks. Groups such as the World Health Organization and the International Summit on Human Genome Editing provide platforms for cross-border collaboration and consensus building. These forums encourage transparency, data sharing, and adherence to principles such as beneficence, non-maleficence, and justice. The goal is to create a shared foundation that supports innovation while safeguarding human rights and environmental health. Such global coordination helps to mitigate risks associated with divergent national policies and ensures that the benefits of gene editing technologies can be realized broadly without exacerbating inequalities or ethical dilemmas. As the regulatory landscape continues to take shape, it remains clear that balancing innovation with safety is an ongoing process, one that requires vigilance, flexibility, and open dialogue. The diversity of approaches around the world reflects varying historical, cultural, and social contexts, reminding us that the governance of gene editing is not a one-size-fits-all endeavor. Instead, it is a delicate dance that calls for cooperation between scientists, policymakers, ethicists, and the public. In this shared journey, we learn to appreciate the profound responsibility that comes with wielding such powerful technologies and the imperative to guide them with wisdom and care. Among the many promising applications of gene editing technologies, CRISPR stands out as a transformative tool in the fight against infectious diseases. By harnessing the precision of CRISPR-Cas systems, researchers have begun to explore new ways to target viral genomes with unprecedented accuracy. This approach offers the potential to disrupt the life cycle of viruses within infected cells, thereby reducing viral loads and limiting disease progression. One of the most studied examples is the effort to combat HIV, a virus that has long eluded a definitive cure. Through careful design of CRISPR molecules that specifically recognize and cleave HIV DNA integrated into human cells, scientists are making strides towards strategies that could eventually eradicate latent viral reservoirs. Such work is emblematic of a broader vision, where gene editing may provide not only treatments, but also preventative measures that can be deployed rapidly in response to emerging viral threats. The elegance of CRISPR's mechanism lies in its adaptability. Researchers can program the system to seek out genetic sequences unique to a given pathogen, allowing for targeted intervention without harming the host's genome. This specificity reduces the risk of unintended side effects, a critical consideration when manipulating complex biological systems. Beyond HIV, CRISPR-based strategies are being investigated for their potential to neutralize a variety of viruses, including those responsible for hepatitis B, human papillomavirus, and even the SARS-CoV-2 virus that caused the recent global pandemic. In laboratory settings, scientists have demonstrated the ability to degrade viral RNA or DNA, effectively halting replication and spread. These early successes hint at a future where gene editing could complement traditional antiviral drugs and vaccines, offering a versatile toolkit to respond to infectious diseases more swiftly and effectively. The path forward involves meticulous research, rigorous testing, and thoughtful consideration of ethical implications to ensure that such interventions are both safe and equitable. In parallel with CRISPR, other gene editing technologies continue to evolve, expanding the possibilities for infectious disease control. Techniques such as base editing and prime editing offer refined methods to correct or disrupt genetic material with even greater precision and reduced off-target effects. These advancements may further enhance our ability to engineer immune cells that can better recognize and eliminate pathogens, or to develop gene drives that could help control vector-borne diseases like malaria. As these technologies mature, interdisciplinary collaborations between molecular biologists, clinicians, bioinformaticians, and public health experts will be essential to translate laboratory findings into practical therapies and preventive measures. The integration of gene editing into infectious disease research exemplifies the intersection of innovation and responsibility, where each step forward is guided by a commitment to improve human health while respecting the complexity of living systems. While the promise of gene editing to combat infectious diseases is profound, it also invites careful reflection on the challenges that lie ahead. The delivery of CRISPR components into the right cells at the right time remains a technical hurdle, as does the need to avoid unintended immune responses or long-term consequences. Moreover, the equitable distribution of these revolutionary treatments poses questions that extend beyond science, touching on issues of global health justice and access. As research progresses, it will be vital to engage diverse communities in dialogue, ensuring that the benefits of gene editing technologies reach all populations and do not exacerbate existing disparities. Navigating these complexities requires patience, humility, and a steadfast dedication to ethical principles, as the scientific community continues to explore this new frontier. As the field of gene editing continues to evolve, researchers have developed next-generation techniques that offer remarkable precision and reduced risks compared to earlier methods. Among these advancements are base editing and prime editing, innovative approaches that enable direct, targeted changes to DNA sequences without creating double-strand breaks. This is significant because traditional CRISPR-Cas9 editing
relies on cutting both strands of the DNA helix, which can sometimes lead to unintended mutations or chromosomal rearrangements. Base editing, in particular, allows for the conversion of one DNA base pair into another with striking accuracy, effectively rewriting single letters in the genetic code. This capability opens new avenues for correcting point mutations that underlie many genetic diseases, doing so with minimal disruption to the surrounding genome. The elegance of base editing lies in its simplicity and specificity, which together reduce the likelihood of off-target effects and cellular stress.
Complementing base editing is prime editing, a more versatile technique that extends the range of possible genetic modifications. Prime editing employs a fusion of a catalytically impaired Cas9 enzyme and a reverse transcriptase, guided by a specialized RNA sequence that both identifies the target site and carries the desired genetic correction. This system can introduce insertions, deletions, and all 12 possible base-to-base conversions, offering unparalleled flexibility in rewriting genetic information. Unlike conventional CRISPR, which depends on the cell's error-prone repair mechanisms following DNA cuts, prime editing programs the desired edit directly into the DNA strand, improving precision and reducing unintended consequences. Early laboratory studies have demonstrated the potential of prime editing to correct mutations associated with sickle cell anemia, Tay-Sachs disease, and other inherited disorders. The continued refinement of prime editing holds promise not only for therapeutic applications, but also for advancing our understanding of gene function and regulation. While these emerging technologies represent significant strides forward, they are accompanied by new challenges that researchers must carefully address. Efficient delivery of base editors and prime editors to specific tissues remains a central obstacle, as does ensuring their activity is tightly controlled to prevent unintended editing events. Moreover, the long-term effects of such precise genome modifications are still being explored, requiring extensive preclinical and clinical studies to establish safety and efficacy. Ethical considerations continue to play a vital role, particularly as these tools make it conceivable to edit human embryos or germline cells in ways that could be inherited by future generations. Engaging in thoughtful dialogue with ethicists, policymakers, and the public will be essential to navigate the responsible development and application of these powerful technologies. As the scientific community advances, a balanced approach that weighs innovation with caution will guide the path forward. The future of gene editing is not limited to base and prime editing alone. Other emerging modalities, such as epigenome editing and RNA-targeted techniques, are expanding the possibilities for modulating gene expression without altering the underlying DNA sequence. These methods offer reversible and dynamic control over biological processes, potentially allowing for treatments that are both precise and adaptable. Together, these converging technologies form a rich landscape of tools that can be tailored to address a wide range of medical and agricultural challenges. As research deepens, the integration of these approaches may lead to sophisticated therapies that not only correct genetic defects, but also enhance resilience and adaptability in complex biological systems. The horizon of genome engineering is broadening, inviting new perspectives on how we might shape the future of life itself.
At the intersection of gene editing and synthetic biology lies a profound opportunity to reimagine life's blueprint. Synthetic biology, with its emphasis on designing and constructing new biological parts, devices, and systems, complements the precision of gene editing tools like CRISPR. This synergy allows scientists not only to modify existing genomes, but also to assemble entirely new genetic circuits that can perform novel functions. By combining these approaches, it becomes possible to engineer organisms with capabilities never before seen in nature. Microbes that produce sustainable biofuels, plants that fix nitrogen more efficiently, or cells engineered to sense and respond to their environment in programmed ways. Such innovations mark a shift from simply reading and rewriting DNA to writing new chapters in biology's story. The careful orchestration of gene editing within synthetic frameworks offers a way to expand our toolkit beyond natural constraints, opening the door to bespoke organisms tailored for specific tasks. One of the most compelling visions emerging from this integration is the concept of biofactories, living systems designed to manufacture valuable compounds, medicines, or materials at scale. Through precise gene editing, pathways within microorganisms can be optimized or rewired, enabling efficient production of complex molecules that might otherwise be difficult or costly to synthesize chemically. Synthetic biology contributes modular components and standardized parts that streamline the design process, allowing iterative testing and refinement. For example, engineered bacteria or yeast can be programmed to produce insulin, bio-based plastics, or even novel antibiotics, potentially transforming industries and improving sustainability. Such biofactories represent a harmonious blend of natural efficiency and human ingenuity, where life itself becomes a platform for manufacturing with minimal environmental footprint. Beyond industrial applications, the fusion of gene editing and synthetic biology holds profound implications for medicine and environmental stewardship. Custom-designed cells could one day be deployed as living therapies, engineered to detect disease markers and deliver targeted treatments within the body. Alternatively, synthetic organisms might be created to break down pollutants or restore ecological balance in damaged habitats. These advances rely on the ability to precisely control genetic information and cellular behavior, a challenge that gene editing tools are uniquely suited to address. By constructing synthetic networks of genes that respond predictably to external signals, researchers can imbue cells with new layers of intelligence and adaptability. This level of control heralds a future in which biological systems are no longer passive, but active participants in solving complex health and environmental challenges. The journey toward these possibilities is guided by an evolving understanding of genetic regulation, cellular communication, and systems biology. As synthetic biology matures alongside gene editing, it encourages a holistic approach, designing organisms from the ground up while respecting the delicate balances inherent in living systems. Ethical considerations and safety measures are integral to this process,
ensuring that engineered life forms serve humanity without unintended consequences. The iterative cycles of design, build, test, and learn create a dynamic feedback loop, fostering innovation while maintaining responsible stewardship. In this way, the combined power of gene editing and synthetic biology is shaping a future where life can be sculpted with unprecedented nuance and purpose.
The advent of gene editing technologies, notably CRISPR-Cas9, has opened a new chapter in the realm of personalized medicine. This approach recognizes the unique genetic makeup of each individual and aims to tailor medical treatments accordingly. By precisely targeting specific genes that influence disease susceptibility and drug response, gene editing offers the promise of therapies that are not only more effective, but also minimize adverse effects. Instead of applying a one-size-fits-all model, personalized medicine, guided by gene editing, can adjust interventions to the subtle nuances of an individual's genome, unlocking a level of precision previously unattainable. This shift is particularly profound in complex diseases such as cancer, where genetic variations dictate how tumors evolve and respond to treatment. Beyond CRISPR, emerging gene editing tools like base editors and prime editors refine the ability to make subtle, accurate changes in DNA sequences without causing unintended breaks. These advancements enhance the safety profile of gene therapies and expand the range of genetic conditions that can be addressed. When integrated with comprehensive genomic sequencing and bioinformatics, these technologies enable clinicians to design therapeutic strategies that consider the entirety of a patient's genetic landscape. This holistic perspective supports the development of treatments that are finely tuned to counteract the underlying genetic causes of disease, moving beyond symptomatic relief toward lasting cures. The interplay between a patient's genome and their environment can also be better understood, allowing for dynamic adjustments in treatment plans as new information emerges. The potential applications of personalized gene editing extend beyond treating inherited disorders to include infectious diseases and even age-related conditions. For example, editing genes that regulate immune responses could enhance the body's ability to fight infections or modulate inflammation, a key factor in many chronic diseases. Additionally, as research uncovers genetic markers associated with longevity and healthy aging, gene editing may one day contribute to strategies that promote resilience and vitality over a lifetime. The integration of gene editing within personalized medicine thus represents a convergence of cutting-edge science and compassionate care, emphasizing treatments that honor the individuality of each patient. As these technologies continue to evolve, they invite ongoing dialogue about ethical frameworks and equitable access, ensuring that the benefits of personalized gene editing can be realized responsibly. As personalized medicine powered by gene editing advances, it also transforms research methodologies, fostering a more iterative and patient-centric approach.
clinical trials can be designed to assess treatments in the context of specific genetic profiles, improving the accuracy of outcome predictions and accelerating the development of new therapies. This data-driven refinement creates a cycle of continuous learning, where each patient's response informs future interventions. Moreover, the convergence of artificial intelligence with gene editing facilitates the analysis of vast genomic datasets, identifying patterns and potential targets that might otherwise remain hidden. Together, these innovations contribute to a future where healthcare is not only more precise, but also more adaptive and responsive to the evolving understanding of human biology.
In this evolving landscape, the collaboration between researchers, clinicians, and patients forms the foundation of progress, ensuring that gene editing technologies are applied with care and insight. The path ahead invites us to imagine a world where treatments are crafted with the same individuality as the people they serve, where the genetic intricacies that define us become tools for healing. As we explore these possibilities further, the horizon of personalized medicine continues to expand, inviting new questions and inspiring innovative solutions that honor the profound complexity of life itself. As we venture deeper into the realm of gene editing, one of the foremost challenges lies in the delicate art of delivery. The molecular tools, such as CRISPR-Cas systems and other gene editors, must reach their intended destinations within the body, specific cells that require correction or modification. This task is far from simple. The human body is a complex network of tissues and barriers that can impede the journey of these therapeutic agents. Researchers have explored various delivery vehicles, including viral vectors, lipid nanoparticles, and novel biomaterials, each with its own set of advantages and limitations. The goal remains consistent: to transport gene editors efficiently and precisely, minimizing off-target effects and avoiding unintended consequences. Successfully navigating this biological landscape requires a profound understanding of cellular environments and the mechanisms that govern molecular uptake. This ongoing quest to perfect delivery systems is pivotal, as it determines not only the efficacy of treatments, but also their safety and scalability for widespread use. Alongside the challenge of delivery, the immune response presents another intricate hurdle in the path of gene editing therapies. As foreign molecules enter the body, the immune system may recognize these gene editors as threats, initiating a defense that can neutralize their therapeutic potential. This reaction can manifest as inflammation or the production of antibodies, which may reduce the effectiveness of treatment or lead to adverse effects. The immune system's vigilance, while essential for protecting us against pathogens, complicates the administration of gene editors derived from bacterial sources, such as the Cas proteins used in CRISPR technology. To address this, scientists are investigating ways to cloak these tools or engineer them to be less immunogenic. Strategies include modifying protein structures, using transient delivery methods, or harnessing immune-suppressing agents to create a more hospitable environment for gene editing.
This delicate balance between evading immune detection and preserving the body's natural defenses is a critical frontier in the development of safe and lasting genetic therapies. The interplay between delivery and immune response underscores the complexity of translating gene editing from the laboratory to clinical practice. Each patient presents a unique biological landscape, with variations in immune profiles and cellular architecture that influence how therapies perform. Personalized approaches to overcoming these obstacles are emerging, where treatments are tailored not only to the genetic condition, but also to the individual's immunological nuances. This personalized strategy demands a convergence of disciplines, blending molecular biology, immunology, and bioengineering to craft solutions that harmonize with the body's intricate systems. Moreover, ongoing clinical trials continue to shed light on the long-term implications of gene editing interventions, informing safer and more effective protocols. These endeavors reflect a profound respect for the balance between innovation and caution, recognizing that the promise of gene editing is intertwined with the responsibility to protect and preserve human health. As research advances, emerging technologies are also being explored to complement and enhance current gene editing methods. For instance, novel delivery techniques such as extracellular vesicles and engineered bacteria offer intriguing possibilities for more targeted and less immunogenic transport. These innovative approaches aim to overcome the physical and biological barriers that have long challenged the field. Simultaneously, the refinement of gene editors themselves through improved specificity and reduced size helps to lessen immune recognition and improve cellular uptake. The dynamic evolution of these technologies exemplifies the creative spirit driving modern science, continually seeking new pathways to unlock the therapeutic potential hidden within our DNA. Each step forward brings us closer to a future where gene editing can be applied with subtlety and precision, embodying a harmony between human ingenuity and the wisdom inherent in living systems. Among the most remarkable strides in gene editing are recent clinical trials demonstrating the true potential of CRISPR as a therapeutic tool. In 2020, a landmark study published in Nature Medicine detailed the first successful use of CRISPR to treat an inherited form of blindness known as Leber congenital amaurosis. This condition, caused by mutations in a single gene, results in severe vision loss starting in infancy. Researchers employed CRISPR to precisely edit the faulty gene directly within the patient's retinal cells, a delicate and challenging task given the eye's complexity and sensitivity. The results were encouraging, revealing not only restored genetic function, but also measurable improvements in vision. This trial marked a pivotal moment, showing that gene editing could move beyond the laboratory and into the clinic, offering hope for treating previously intractable genetic disorders. Such progress underscores the meticulous efforts behind refining delivery methods and ensuring safety, which are fundamental to realizing gene editing's promise. Beyond CRISPR, a variety of emerging gene editing technologies are shaping the landscape of genetic medicine with their unique advantages.
Base editors and prime editors, for example, allow scientists to make precise single-letter changes in DNA without creating breaks in the genetic code. This reduces unintended consequences and increases the safety profile of gene editing interventions. Recent studies have utilized these refined tools to correct mutations responsible for blood disorders such as sickle cell anemia and beta thalassemia, with several clinical trials now underway. These approaches not only expand the therapeutic scope, but also demonstrate how incremental improvements in molecular engineering can translate into powerful applications. The continuous refinement of gene editors illustrates a broader theme in science, the quest for greater precision and control, enabling interventions that are both effective and gentle at the cellular level. The future of gene editing is further enriched by advancements in delivery technologies that can ferry molecular tools safely and efficiently to target cells. Innovative methods such as extracellular vesicles derived from stem cells offer biocompatible carriers that reduce immune activation and improve cellular uptake. Engineered bacteria capable of homing to specific tissues present another intriguing avenue for targeted delivery. These biological vehicles can potentially overcome the limitations posed by traditional viral vectors, which sometimes induce immune responses or have cargo size restrictions. Recent experiments have demonstrated successful gene editing in animal models using these novel delivery systems, providing a glimpse into more precise and patient-friendly therapies. These innovations highlight the intricate interplay between biology and engineering, where understanding natural processes inspires the design of advanced therapeutic platforms. Intriguingly, ongoing research is also exploring the ethical and societal dimensions accompanying these technological advances. As gene editing moves closer to widespread clinical use, scientists and ethicists alike are engaging in dialogue to ensure responsible stewardship of these powerful tools. Discussions focus on balancing innovation with caution, particularly when considering heritable genome editing and applications beyond therapeutic needs. This conscious reflection emphasizes the importance of transparency, inclusivity, and long-term monitoring in the deployment of gene editing technologies. It is a testament to the scientific community's commitment to not only advancing knowledge, but also honoring the profound responsibility that comes with altering the very fabric of life. Such a holistic approach enriches the foundation upon which future breakthroughs will be built, weaving together technical mastery with ethical mindfulness. Together, these exciting research highlights paint a vivid picture of a rapidly evolving field where precision, safety, and thoughtful innovation converge. Each study, each trial, and each technological refinement adds a new thread to the tapestry of gene editing, bringing us closer to harnessing this extraordinary capability for the benefit of all living beings. As we continue to explore these frontiers, the horizon broadens, inviting us to imagine a future
where genetic diseases may be corrected at their source, where personalized medicine becomes the norm, and where life itself can be understood and nurtured in ever more profound ways. Building on this momentum, the next chapters of scientific inquiry will delve deeper into the integration of gene editing with other emerging disciplines, opening new pathways for discovery and healing. As we stand on the threshold of a new era, gene editing technologies like CRISPR offer a gentle promise. The ability to rewrite the very code of life with precision and care. Imagine a world where inherited diseases such as cystic fibrosis, sickle cell anemia, and Huntington's disease are no longer a source of suffering or heartbreak. By targeting and correcting the genetic mutations responsible for these conditions, researchers foresee a future where the burden of illness is lifted before it ever takes hold. This vision is not simply about curing disease, but about restoring hope and dignity to countless families around the globe. Through ongoing trials and thoughtful advancements, the possibility of editing genes safely and effectively becomes less a distant dream and more an attainable reality. The delicate balancing act of refinement and responsibility guides every step, ensuring that the power to alter life's blueprint is wielded with the utmost respect for ethical boundaries and human diversity. Beyond human health, gene editing holds transformative potential for the natural world and the sustainability of our planet. Agricultural innovations rooted in gene editing techniques promise crops with enhanced nutrition, resistance to pests, and resilience to climate change. By fine-tuning the genetic makeup of staple foods, scientists envision fields where drought-resistant grains flourish and harvests increase without the heavy reliance on chemical fertilizers or pesticides. This shift could greatly improve food security, especially in regions vulnerable to environmental stress and population growth. Such advancements extend far beyond yield. They represent a harmonious collaboration between technology and nature, designed to nurture the earth while feeding its inhabitants. As we explore the possibilities of gene-edited plants and animals, the goal remains clear: to cultivate a world where nourishment is abundant, sustainable, and accessible. The integration of gene editing into medicine and agriculture is only the beginning of a broader transformation that may redefine how we relate to life itself. Future therapies may become increasingly personalized, crafted to match the unique genetic profiles of individuals, reducing side effects and improving outcomes. Simultaneously, gene editing tools will likely be combined with advances in artificial intelligence, synthetic biology, and regenerative medicine, creating unprecedented opportunities for healing and innovation. In this unfolding landscape, the capacity to edit genes might extend to restoring damaged tissues, combating aging, or even preserving endangered species by correcting harmful genetic variations. Each breakthrough will require careful deliberation, as the ethical dimensions of altering life's foundation invite us to consider our shared responsibilities and the legacy we leave for generations to come. This unfolding journey invites us to reimagine the relationship between humanity, nature, and technology in a spirit of stewardship and hope. As we stand on the threshold of a new era shaped by gene editing technologies like CRISPR, we are reminded that with great power comes profound responsibility. These tools offer extraordinary potential to alleviate suffering, enhance health, and protect our environment. Yet they also raise complex ethical questions that cannot be overlooked. The ability to alter the very code of life invites us to reflect deeply on what it means to intervene in natural processes. It is essential that the scientific community, policymakers, and society at large engage in thoughtful dialogue to establish clear guidelines and safeguards. This will ensure that the remarkable promise of gene editing is realized in ways that respect human dignity, biodiversity, and the delicate balance of ecosystems. In doing so, we honor not only the science itself, but the values that define our shared humanity. Responsible innovation calls for a harmonious blend of curiosity and caution. As researchers push boundaries, transparency and inclusivity must guide the development and application of gene editing technologies. It is important to foster international collaboration and equitable access so that benefits are shared broadly rather than concentrated among a few. Ethical stewardship also means anticipating unintended consequences, investing in long-term monitoring, and remaining open to revising approaches as new knowledge emerges. By embedding ethical reflection into every stage of innovation, from laboratory research to clinical use and environmental release, we nurture a culture of care that prioritizes the well-being of current and future generations. This mindful approach not only mitigates risks, but cultivates trust, an essential foundation for progress in any transformative field. In embracing gene editing as a force for good, we are called to imagine a future where technology uplifts life in all its forms, without compromising the integrity of natural systems. This vision invites us to consider the interconnectedness of human health, ecological resilience, and social justice. By weaving ethical principles into the fabric of scientific discovery, we create pathways for innovation that are both bold and compassionate. It is through this lens of stewardship that we can responsibly harness gene editing to address pressing challenges such as genetic diseases, food security, and environmental degradation. Each step forward becomes not merely a technical achievement, but a deliberate act of care for the planet and its inhabitants. In this way, the story of gene editing is also a story of hope, a testament to human creativity, guided by wisdom and empathy. As we conclude this reflection on the transformative potential of gene editing technologies, it is clear that their impact will be shaped as much by our choices as by our discoveries. The future of life, in all its diversity and complexity, rests in our hands. By embracing responsible innovation grounded in ethical stewardship, we honor the profound gift of knowledge while safeguarding the delicate balance of life itself. In this shared journey, we are reminded that science is not only a tool for understanding the world, but a means to nurture it with care and respect. Let us move forward with humility, thoughtful intention, and a hopeful spirit, confident that together we can shape a future where the promise of gene editing enriches life without compromising the values that connect us all.